Welcome back, everyone. So the next topic I wanted to cover in today's lecture is holography. So you might have seen holograms or holography in movies and kind of in a lot of, so for example, Star Wars, like there's Princess Leia in the New Hope. So this has been famous and well known for a long time. So what is the difference between an image and a hologram? So one kind of way to think about this is, let's say you have take a picture, a photograph of using a camera and print it out on paper and keep it in like life-size object. How is that different from a person actually standing next to this? So what is happening is that when you take an image, you take the magnitude square. So the phase information is essentially lost. And so what a hologram does is, apart from having an intensity profile of the object, it has the phase of the object. And so the combination of the intensity and the field and the phase together give you the three-dimensional structure of the object. What if, like the basic difference between a cardboard cutout of a person and like a picture, if you take a photograph and print it, and a like something which is realistic, is the three-dimensional nature of it. Okay, so what holography essentially does is tries to replicate the field, the light that comes towards you. Okay, so what do I mean by that? Means that, let's say you have an object, light comes from, like light falls, like the light, like this light bulb shines on the person, and then the light hits your eyes. Then you see the person. So now what you see depends on the light. All you can see is the light that reaches your eyes. If the nature of the wavefront that reaches your eyes is exactly the same as like what is getting emitted because of the person standing or because of something else, it will be indistinguishable for you. Okay, what I mean is, like let's say you have a wavefront coming towards your eyes. It could be created by like a screen, or it could be created by a person. If the exact shape of the wavefront in both cases is the same, then you will not know the difference between the real life thing, real life person, and something which has been created. Okay, and if we can create this wavefront shape, so that means wavefront shape includes the magnitude and the phase. So if you can figure out both the magnitude and the phase, then you can create a hologram. That's the essential idea of holography. Okay, so you want to record both the magnitude and the phase, and if you can do that, then you can kind of create realistic renderings of anything that looks like 3D objects. That's essentially the idea. The question of how to do it first figured out by Gabor, like a scientist by the name Gabor, in the mid 19th, like about in 1950 or so, ballpark. And so these are few examples, like sort of, if like you can see a mouse, you can get a hologram of a mouse, and you can, like, if you look at the hologram, basically it looks like what looks like a photo, which is typically two-dimensional, but a hologram will, if you move your eyes, it looks 3D. And it's really not 3D, but it looks 3D because the wavefront of light that is coming from this particular object essentially is identical to what you would see if there was actually a mouse there and a mouse is standing at that. So similarly, like these days, you can people have, like, if you want to see visualize say some brain or something that's not in 3D, then you can actually create a hologram, or even in kind of like for like a dance setting, like different kind of, you can make an object in 3D look and like thing like that in front of you. Or if you go to a museum, you can have planets around you which look like realistic in 3D. And all these, how are they made? They are made with holography. Okay, newer versions of holography. And in the next few minutes, I want to talk about exact how does holography work. the basic principle, how to make one, how like kind of what's the basic idea behind this and so on. Okay, so here is the essence of what we need to do. Okay, so that which we want to record. So what you do is, like this object, keep it on one side, and then take what's called a reference light. Okay, so what you do is take the object, light from that comes like goes in all directions, and let's say it comes towards this way too. And then you have a reference beam. You interfere the two beams, and on this plate, on what is called a holographic plate. Okay, so now what happens is that the light that goes afterwards will basically is a combination of this light and this light. So some amount of light, like after interference, goes through out here, and if you put your eye here, you can see this object over here. Now what you do is remove the object and still have this reference beam, and then have this grating kept here. So this recording basically has recorded light that comes because of the object. So it's this kind of a plate. It's like a photographic plate. You know, if you remember your old-timey photographic films, like now there are newer versions of this, but basically it's like a photopolymer. When light falls on it, it polymerizes, it becomes dark and light in the correct places. So it's like a record of the field, the light intensity that is falling over here. You're interfering these two. If light falls on this, it scatters and light is emitted in this direction. And if you put your eyes here, if you see from this side, you will see what looks like the object. The nice thing about this is that even if you remove the object, once this photographic plate or the holographic plate is developed, then if you remove the object and just shine the reference beam, light will still scatter. The wavefront that you get out is the same as before. So previously, there is an object which is emitting some kind of a wavefront and you see the wavefront. Now you are creating a plate, which is making a plate here, which is such that when light falls on it, it also emits the same kind of light as before. And so because the wavefront that reaches your eyes is exactly the same as before, it looks like the 3D object that was previously there. That's the essence of holography. Okay, so the question, plate and why does it work? Okay, so that's the next thing we'll discuss. But this is like, any questions about the basic idea? Okay. Yeah. So here is how it essentially works. Okay. So this is what access holography. So light is coming in one direction here, and you combine the two, and this is, yeah. So this is the light from the object. This is the light from the reference. You add, like here they interfere, right? Like add and take the magnitude square. Intensity gets recorded. So take U0 plus UR squared, UO plus UR squared. And this becomes first term plus second term plus 2 cross terms. So intensity of the reference, intensity of the object, and then you have 2 cross terms. It is terms that are crucial in holography. This is really what creates the hologram. So the intensity of these two are so this term is basically the angle of the phase because of the reference minus the phase because of the object itself. So this captures, so I0 IO here essentially is the intensity of the object, and here you have the phase of the object. So the what is recorded on this holographic plate is basically it has information about the intensity and the phase, and therefore when you have both the intensity and the phase recorded and you shine light on it, it creates a wavefront on the other side, and that wavefront will be exactly the same as the light coming, wavefront comes from the object. And this how you basically it makes it look like the object is actually there in 3D dimension. So yeah, so this is the process of recording. And then do a reconstruction, meaning if you remove the object, then how does it work? Like, what do you need to do to make it look like this? You just have to have like the reference arm continue, reference beam continue to be there. Okay. Okay. So in this case, T here is the recording, the holograph recording. In the construct, in other words, visualizing how the hologram looks like, take T multiplied by U of R. You have take the holographic film, on that have the reference arm, reference beam UR incident just like before. And so what you have is multiplication of T times UR. If you multiply the two, you will get this kind of a field. And this, as you can see, has 0 of x comma y. And this means basically the field that is instead of previous. Sorry, can you hear me? Yeah. So if you just take a picture, if you take a camera and take a picture, what you will record is I0 of x comma y. So the intensity is measured, the phase is lost. But in holography, you get U0 of x comma y. So the field, so U0 is a complex number. It has the magnitude and an angle. So the angle has the phase information. And so this way, you have the magnitude and phase going towards like in the, and if you stand in this direction and take a look at it, then you will see the field as the exact shape of the wavefront that you will get. So this is like a mathematical way of saying that you can see like this is how it works. This is how you see holography, a hologram if you stand in this direction. But from this equation, you may have noticed something else as well. Of course, there is intensity, which is there from before. There is an extra term, which is U star or U conjugate. It's the complex conjugate of U naught of x comma y. That also is there. It's like ideally you just want it to be U0 of x comma y and nothing else. That's when you see a perfect hologram. But in practice, what you see is that you see U0 of x comma y and U0 conjugate of x comma y. So that is what's called so that is basically in some sense a fake image. It's a second image of the object which you also see necessarily. And this is like it's called the conjugate, which it's called the conjugate, which is something which happens in holography, which is not easy to avoid. There are ways of dealing with it, which I'll talk about. But this is one of the kind of issues with holography. You can look at it only if you look at it only from this angle, it looks perfectly fine. But if you look at it from a slightly different angle, you may be seeing the mixture of the object and the complex conjugate, and that's when it will not look exactly right. So holography of this type, off-axis holography, works very well when you look only for you look at it from a certain angle or a certain range of angles. If you look if you go on the other side, so if you have a 3D object, you can go around behind it and see, right, and you can still see, right? But the limitation of holography of this sort is that because of this complex conjugate problem, only a limited range of angles allow you to see the object in 3D. If you move in a direction, or at least move far away at a much larger angle, instead of seeing it from here, if you see it from here or see it from here, then there be some problems. Okay, so that's a limitation of this sort of holography, and this is called complex conjugate or the conjugate problem of holography. So after you do the recording, if you have a reference beam here, so the equation here that I mentioned, says that you have intensity of light, which is basically going to straight like this. Then the U0 field goes this way, and the U0 conjugate goes downward. So the real field goes upwards, and the conjugate field goes downwards, and that is kind of the regular the U0 I0 field goes this way. So that's how it like that's so yeah. Yeah, so you can see the kind of problem you will encounter. Okay, yeah. So so there are few ways to deal with it. One is sure like sorry, I'm hearing some background noise. Are you able to? Keep going. Okay, yeah. All right. So one way to deal with it is basically only look at the hologram from a certain range of angles. So if you look at it from this set of angles, you will have ambiguity. You can see from this angle or you can see from this angle, but if you see from somewhere in between, it will not be or it will have some issue. So that's one one simple way to deal with like this problem is that you can basically look at it from the right set of angles. Another way to deal with it is using a Fourier approach. So you can use a complex Fourier kind of a filter for holography. So what do I mean by that? Instead of making a recording of the object itself, what you do is take the object, put it in front of a lens and at a distance f, and another distance f. Basically, in other words, you take the Fourier plane of the object and do a recording, take a reference and have it incident on the Fourier plane. So instead of getting a hologram of the object, you get a hologram of its Fourier transform. That's another way to kind of deal with this problem. And so when you do that, you're recording the Fourier transform coefficient on the hologram. Instead of recording the object shape, you're recording the Fourier coefficient. Now, if you have light incident, reference arm incident, and you put another lens here, then you remember you have through one lens at a distance f and another distance f. So what will you get? You essentially get the inverse of the object, right? Like the inverted object. That is what you will see on this. But if you don't put a lens on this side, f conjugate, the conjugate part, you can let it go. Because it's in the Fourier domain, you will not see it like an object. So only when you the second lens will make a real object, the object out of the first one, but you will ignore the second part. And in that way, you can basically get a good holographic image without having the other complex components interfere with the conjugate components interfere with your real part. So that is another way. Using a Fourier approach, you can get a good hologram and avoid the complex conjugate problem by using this kind of approach. Okay, yeah. So this is so this kind of an approach is called Van der Lubbe filter, holographic filter, and this is so this is this is also commonly used. Any questions? Yeah. Why the reference? Okay, yeah. So that's that's an interesting question. So whenever you have like back to this figure, okay. So when you are doing it, you interfere the reference with the object. Then, in order to what you have the recording on the plate is the intensity T, the T. And if you just have T, then you need some light to illuminate it so that you see something on the other side. Where if you just have a holographic film, then there is no light incident. It does not emit light by itself. It's just like a black and white pattern of some sort. Only if you have some other new light incident. So if you multiply, like you have UR reference field incident on it, then that will produce a hologram. So you should have light in the back, which is the same as the reference what was there in the reference. Yeah, correct. Yes, whatever when you were creating the hologram, whatever reference light was there, you keep it on, or you can like sort of when you turn that on the holographic plate, then you will create the hologram. It's a combination. So basically, it's a combination of the T, the holographic recording, times U of R, which is the reference light. Mm-hmm. Exactly. You can, in fact, so you might have seen these kind of different kind of holographic, like kind of holography, like you use stickers which look 3D and stuff, right? So there are several, like so there are slightly newer technologies. So the first previously it was done with this like the film camera. Do you know what a film camera is? It's anyone knows what a film camera is? Maybe you are too young to. Previously, before digital cameras, there was a way of for a long time, for 50 or 60 or more years, so you basically in cameras you put like a basically something like what's called a film, and when you press a button, like the lens aperture would open and would expose this. The light would fall on this photographic film, and you take that film and then give it to somebody who does some chemistry and develop it and then makes a picture out of it. So that actually we used to use silver halide, like it was a chemical which was on this film, which is photosensitive. And so I guess all of you are too young to know what a film is, but okay. So basically, that was how you could. So it is something which is where if light falls on it, there is a chemical reaction that takes place, and it becomes dark, or it changes the chemical. Like it becomes a bit dark where there is more light where it falls. It becomes darker, and that basically it's like that kind of. It's a photosensitive material. Okay. So now you also have other kinds of materials which what are called photopolymers, where if you it's the same principle, but like sort of you don't need to put it in do additional processing. You don't need to send it off to some chemical lab where they kind of dip it in something and make a kind of picture out of it. So like you just have light incident, and wherever high intensity is incident, there it becomes darker. Wherever low intensity, it still continues to be transparent, and so basically you can that's the kind of and some places you can use what's called photoresist. So it's the same type of process which is used for making like your electronics, electronic circuits and so on. So the nanofabrication people use what's called a photoresist. So you have UV light or some light incident wherever light is incident, it polymerizes. Then you wash it away and create kind of like a electronic circuit. All electronic chips, for example, Intel and like NVIDIA and like those like kind of people who make chips use the same basic idea. So you kind of they also use something like photolithography, something of that. So you can use the same process as well to do this. And once you have one of these, you can make it out of metal and put it on, press it onto plastic sheets and basically make 100 of these holograms, holographic plates. So once you have one holographic recording, there is a process by which you can make almost like printing. It's like heat it, press it like this and remove it, and it will make a holographic plate. And then you shine light and you can make 1,000 of these. And like you can have holograms, like light, as long as light is incident, it will give you a hologram on the other side. so you can get these. Any other question? Okay. All right. So this is kind of how how the recording is actually made. Typically, the reference and the object, you use one laser or the same beam to kind of create an interference. So if you have two different beams, we will say later that you cannot have them interfere. There's no phase relation. So if you have the same beam, use a beam splitter and split it into two parts. One is the reference beam, which is incident on the holographic plate. Other one is incident to the object, and it shines, scatters and reflects light, and these two interfere, and you do a recording over here. And then later on, you just keep the reference as before, just remove the object, and whatever light comes out will be exactly the same as what you would see when the recording took place, and it looked like it's 3D. This is essentially how you can make a holographic recording. Yeah, so and this is actually how what is recorded on the plate looks something like this. If like say the letter P, like if the object is in the shape of a letter P, then you can, like, this is like the holographic record. It really doesn't look anything like the letter P because it's an interference pattern which is a combination of like a Fourier transform of the object and kind of the reference arm combined together, and then it's just like sort of so this is the recording. And but when you shine a light on this, what you see at the other end looks something like this. Okay, so in fact this is the actual image, and then this is the complex conjugate, and this is the DC, the intensity I of the XY, right? So that's what you see here. The letter P is in fact like you will see it in 3D, and so this is the complex conjugate problem. So if you can isolate this part by blocking out the others in some way, then you can just get a 3D kind of here. So this is the like this illustrates this thing, and then if you can isolate just what you recorded originally, this is how you can do holography. Yeah. That's a good question. So these are different technologies which are used for making holography. Okay, so you can make the one on the right hand side. So the older technologies, when depending on the nature of the setup, like and the chemistry of how exactly what you use to make the hologram, it can be in this way. Yes, it depends on the film. It depends on that how much of exposure, how much time there are a lot of in practice. Whether you say if you are trying to take something as a still object, like something is sitting, then you like say you want to make a recording, you can keep it there and it doesn't. You can keep it exposed for 5 minutes, right? You can take 5 minutes. But if what if something is moving? That it requires, depending on the polymer, like how much time you need to expose to make the holographic film. So it may take a few minutes to now you can make it faster, but if you take a long time and if the object moves, then the recording is messed up, right? So there are all these practical challenges which you have to deal with as an engineer. Like, I'm not going into the details right now, but those are the kind of things. And how much exposure do you need? What light intensity do you need? What is the power of the laser that you're implementing? What is how much reference and how much light should go into should it be 50-50 or something else? Those are all practical questions which you have to deal with. Yeah. Yes, those are experimental aspects which are engineering design specifications essentially. Okay, that is kind of what I wanted to talk about holography. And the next kind of important topic I wanted to talk about is another kind of an imaging system. So I mentioned earlier today that the fact that you can make a face. So what is common between all these systems? the phase, I talked about phase contrast microscopy, I talked about holography. All of them, the crucial thing is they all measure phase. In some way, they figure out what phase the phase of light is. Okay, so you can measure intensity of light, and then you can measure phase of light. If you can measure both, you can get you can get a lot of useful information and make practical systems. Another kind of thing that you can actually do with this kind of a system is what is called quantitative phase imaging. So in the 1930s and so on, so phase contrast microscopy became available, and by 1950s, like a lot of people in biology labs and in other labs were using this phase contrast microscope, and for a long time that was the standard and for the people used. But uh, a few, like after some 40 or 50 years, people thought we should improve phase contrast microscopy. And one of the one of the scientists came up with what is called quantitative phase imaging. So what we showed in phase contrast, you can see a better image, but you could not quantify what the phase is. You can't get phi of x, you don't get a number phi of x comma y, or a set of an image of the phase. You don't get the value of the phase at every given point. So you just get like an image where the phase is like kind of enhanced in some way. Okay. So now the next question was, how could we quantify this? And it took a long time for people to figure out, but in the end, it's not one technology, but it's a bunch of closely related technologies, which is called quantitative phase imaging. So phase imaging is what people did before Zernike and others. The quantitative part is basically what we add just in the past 20 years or so. This has become very important and useful. I'll show you the kind of things which this new phase contrast microscopy or quantitative phase microscopy or imaging allows you to do. So one of the pioneers in this field is a professor by the name Gabriel Popescu. He actually happened to be one of my mentors in my PhD, and unfortunately he passed away recently, but the work he has done is quite phenomenal, and he even wrote a book on this, and some of the things that I talk about is kind of from that, like so let's start with what the imaging system looks like. Okay, so you have a light in a sample, and it goes to an imaging system, and you get an image. And what is the problem? So this is conventional. This is a simple conventional microscope. So the problem with any conventional microscope is that the light incident has magnitude and phase. Because you take the magnitude square, the phase part is lost, and therefore the phi of x comma y is lost in the process of taking the image in a regular microscope. And the point of microscopy is not only figure out that, use the phase, but also find the numerical value of the phase. And that way you can figure out the exact profile, the shape of the refractive index, or shape of the profile of the beam. From that you could derive, say for example, what the refractive index is at every given point, or the optical path length is at every given point, or what is the density of something at any given point, and so on. So there are things that you can study and understand from this kind of a system, which was previously kind of hard to do. So that's the idea. And the way it works is essentially this. Starting from an imaging system, what you do is add a reference. So the principle, the starting point is very similar to Gabor's original idea for holography or Zernike's idea for phase contrast. You have light coming from the sample. You interfere a reference arm with the sample. when you interfere the two, you add them in the fields add, and when take the intensity, when you take the magnitude square, you have the intensity of this original, intensity of the reference, plus these two, and there is a cosine term which has a phi of x comma y. So if you can extract the phi of x comma y quantitatively, then you have quantitative phase imaging. That's the essential idea. How to do this how exactly to do this is the next thing we will talk about. But what are we trying to do? We are trying to find the phase, and in order to find the phase, we have add the or whatever you get from the object, light that comes from the object. You add a reference to it, and then interfere the two, and then so it's an interference-based approach to finding the phase. Any questions? Anyone online? Any questions so far? No. Okay. All right. Yeah. So here is one way to create a quantitative phase image. So what we are interested in is in the phase. In fact, it's more specifically in the phase difference. Whenever you talk about a phase of of light, the thing that you must keep in mind is it's a relative term. If you talk about it's like voltage, right? There is no absolute value of voltage. It's only voltage difference. Potential difference is really all that you can measure with respect to the earth. You can take earth as the ground or like zero or whatever like its voltage is the work done, right? So it's from going from A to B. So it's only the voltage difference that makes like any sense. Similarly, it's the phase so relative to some starting point, light has accumulated a certain amount of phase, and so the relative phase is all that matters. So that's why we with respect to some reference, it could be arbitrary, but with respect to some reference, you are measuring the phase of light. That's something you must always keep in mind. And here, what we are doing is light is incident on the sample. It goes to the imaging system. Apart from the light coming from the sample, what you do is introduce a reference field. You put something like a beam splitter at this point, have a reference beam incident perpendicular. You combine the two and then create an image. So this is take the light from the imaging system, light from the sample which goes to the imaging system, take a reference, add the two, interfere the two, and then capture the image. So this is what is called phase shifting interferometry. So this way, once you do the recording of this UI of xy, you take the magnitude square, you measure the intensity, put a camera sensor there or a detector there, and get an image of the light that light from the system. Then what you get at the end is basically a picture which has both magnitude and phase. But the only thing, this is something which you have we have done before, right? This is what, in some sense, this is a version of what Zernike did in his phase contrast microscopy. What is different now is that you not only take one image for one reference beam, you take 4 different images at 0 degree, you have light of a certain phase incident, shift the phase by 90 degrees, pi over 2 radians, take another image, shift it by 180 degrees, shift it by 270 degrees, and so you get 0, 90, 180 and 270 degrees. You have 4 different images where you shift the phase of the reference by 4 different values. And after that, you get 4 different images, and you can put them all, to combine them through some computational approach, and through this calculation, you can find phi of x comma y. So basically, you will one image alone is not enough to completely get the phase. We need a set of 4 measurements because it's complex numbers. I'll show you the calculation of exactly how to do that. But the idea here is that instead of making one measurement like Zernike did, because we have computed now You have computers and computation as another kind of thing that you can use for microscopy. You introduce light and phase shift the light by different amount and make four different measure, get four different images, and then like do some calculations on them, and that way you get the value of the face. So quantitatively figure out the face at every pixel, and that gives you a map of the entire image. So in fact, so this is kind of the calculation you do. It's fairly simple in this version of you get find the argument. So number, you find an argument of I0 of this image minus this 180 degrees of pi radians image, and this 270 degrees and the 90 degrees image. So you do this and angle calculation. This you get, so basically in the complex plane you get at 0 degree, 90 degrees, 180 degrees and 270, and each of these four images, you combine the two, combine all of them and calculate phi of x comma y, which will give you, after doing this kind of image processing calculation, you get a face of the object at the end of it. Yeah, question? So what's the point of getting the face? Why is it useful? It's something I'll come to in the next, in a few minutes, 5 minutes, okay? But just the fact that the important thing to notice is that it is possible now to find the face through calculations. By doing this kind of set of four measurements and doing some calculations, you can figure out the face of the face profile quantitatively of an object. Question? Okay. Another kind of approach to doing the same kind of, say, finding quantitative face is what is called off-axis interferometry. This is what's called phase shifting interferometry. The second one is called off-axis interferometry. So in this approach, what you do is, instead of light being incident directly like this, perpendicular and incident like this, light you come in at a slightly different angle. So the reference arm is not exactly incident flat, but it incident at a specific angle, a known angle. You have it interfere. Okay, so this is a subtle difference, but an important one. So by doing this kind of an off-axis, so everything else, if I have a sample, it's emitting light which has a certain waveform or phase profile, goes through the imaging system, and before it goes to the detector or camera sensor, you have it interfere with a slightly off-axis, something which is coming from at an angle. You have it incident on the, interfere the two and then have it incident. When you do that, the intense, like you are there a second, the off-axis reference, and then you have a cross curve. So previously, if you remember, you only had like, now you have the phi plus this extra term delta k dot x. Okay, so this is an extra term because you are slightly off axis. So if delta k equal to 0, which is, so delta k tells you what is this new angle compared to the original perpendicular, what angle is coming at. Okay, so if delta k is 0, then you go back to the original version. But in this case, you have coming at an angle, and the good thing, the nice thing about this kind of an approach is that you don't need four measurements. With this one kind of a measurement and some extra calculations, you can figure out the phase. But there are some disadvantages too, which I'll get to. So what you get from this is the cosine term. So you find the cosine term, get the sine term using the Hilbert transform. So there is an extra calculation you can do. So from this measurement, you get cosine of delta kx plus phi of x comma y. From that cosine, there is a relationship between for complex numbers. It so happens that from this cosine term, you can get the sine term using what's called the Hilbert transform. So in fact, this is the equation for the Hilbert transform. And once you get that, you get the cosine term and the sine term. So cosine and sine, so that's the real part and the imaginary part. So you can get the angle of the complex number using the real and the imaginary part, right? So you do that and you can get the phase of the object. One of the problems with this approach is that there is no unique solution in the classical sense. What I mean by that is you have this problem of phase unwrapping. So in other words, if the phase of the sample is something of this sort, then have you heard of the phase unwrapping problem before in any other context? No? Okay. So the basic problem is as follows, right? So you have a complex plane. Let's say a complex number here. So this one, the angle, let's say, is 30 degrees just for now. Okay. When you calculate get the angle to be 30 degrees, it could be 30 or 50 plus 30. This is the same as 360 degrees, 30 degrees, or it could be 720 degrees, 360 times 30 degrees, right? It's the exact same complex number, but the angle of that complex number could be 30 or 360 plus that, or like sort of n times 360 plus that. So basically every that number and its plus multiples of 2 pi or multiples of 360 degrees. So will always, so there is no unique phase value when you calculate the phase. So that's the problem. Okay, so therefore, if the phase, so let's say this is the value of 2 pi or 360 degrees. Even though the phase goes up like this, because the angle you calculate is always between 0 and 2 pi, this phase will come back here. It looks like this purple here and then here. So basically it gets wrapped into the between 0 and 2 pi. The value of the phase will be like 3.5 pi, but that will get wrapped into 3.5 minus 2, that will it will be 1.5 pi. So it will get wrapped in to between 0 and 2 pi always. Okay, so this is what's called the phase wrapping problem. So on how to undo this, you can use there are algorithms to undo it, and you can get this kind of, when you have smooth data like this, you can go from here to here with relative ease. Okay, because it works very well. But when you have noisy data in the real world, when there is noise in the data, did the phase really you cannot know if the phase changed from one point to the next. Did the phase go from 30 to 31 degrees, or did it go from 30 to 361 degrees? You just don't know did it really make a big jump, or was it a small jump and you don't know. So there is ambiguity of some sort. So the real data could be like this, but there is some inherent ambiguity because of this phase unwrapping problem. So that is one of the limitations of this kind of an approach. To show you graphically what it looks like, so this is the intensity if you didn't use kind of quantitative phase imaging. Let's say the data looks something like this. So when you take this intensity and add the reference, then what you recorded the detector, the camera sensor, it looks something like this. Then you take the cosine part, it looks after doing some calculations, it looks something like this. Then if you actually kind of do phase unwrapping, it looks something like this. And then if you remove this kind of the angular, you came in at an angle, so therefore it will create this gradient in the, if you undo this, then the phase looks, after doing all this image processing and signal processing, you'll end up with the face of the object, which has a certain kind of shape and a profile. Okay, so this is how these are some of the important methods. This come under the category of quantitative phase imaging. So there are different versions of quantitative phase imaging. which have each, I told you, phase shifting method, the off-axis method. There are a few other methods as well. So all this tells you the phase profile of the object. So if so the next question you should ask yourself in all these situations is, what's the point? Why did we do this? What is it that we understand about an object that we can, by measuring the phase, what is it that you now know about the object which you did not know before? Okay, that's always, whenever you see anything of the sort, what's the point of doing it? Why are we doing it? Is an important question you always have to answer. Turns out that this quantifying the phase is extremely useful in biology and medicine, and I'll tell you why. Okay, so you can get, so this is the phase contrast microscopy which Wernicke has figured out. So now from this you can go to something like this. This is a quantitative phase image of something else. This is a cell, this is also a pair of two cells. Okay, you can get much more detailed information, and it turns out that you can get detailed information on the nanometer scale of the surface profile of cells using this method. So so traditionally the height of, say you have a cell, if you have a petri dish or a piece of glass on which there is a cell sitting, okay, the height or the thickness of this cell is something which is quite hard to measure. And not only that, not only can quantitative phase imaging measure the height, it can measure it in nanometer, sort of few maybe 10 atoms thickness, sort of you can make it extraordinarily precise measurement of the thickness of this, thickness profile of this cell. Not only that, you can measure it in real time, sort of you can, if the cell is moving and it's kind of changing shape and changing direction depending on the type of cell it is, you can measure how well, how good the properties of the cell are, how the shape of the cell is changing on the nanometer scale. On a very, very high precision scale, you can make measurements of this sort. Okay, so this particular thing happens to be a particular cell, yeah. So this is the phase contrast, Wernicke's phase contrast image, and this is the holographic amplitude, and this is when you're doing some processing, the phase image. This is the kind of phase image you get, and after doing some unwrapping and some phase calculation, this is the kind of thing you get, and this is basically the quantitative phase imaging profile of a couple of cells. Okay, this is one of the things that we do. How is this useful, practically speaking? So here is an example of what you can figure out. So this is it's a red blood cell. So it turns out that a disease like malaria, so what happens is that there is a loss of the shape of, so red blood cells inside the body go or take oxygen and circulate it from the lungs and go to the lungs, get oxygenated, go around the body, and in different parts of the body the oxygen is taken up by different cells, and that's how you get energy. The oxygen is food which becomes glucose and some molecules, and then with oxygen it gets used up inside the cell. That's what gives you energy for your muscles to move on, for everything in the body to go on, right? So oxygen is important, and blood this carries the oxygen to different parts of the body, and it has to squeeze through some capillaries, squeeze through some places which are tiny, and so how well this shape change, so how well, when say the red blood cell, how exactly the shape, what exactly the shape is, how well if you deform it, if you squeeze it and let it come back, does it come back to its original shape? How well does it come back? Does it get deformed? And those kind of things are important properties of red blood cells. And in a disease like malaria, what happens is that its ability to squeeze and come back to its original shape is affected because of the malarial parasite, the consequences of malaria, and how this gets deformed and how it comes back is an important characteristic of the type of malaria it is. So there are it's not just one kind of a disease, there are multiple different types of malaria, and for certain types of malaria, the physical properties of the kind of this membrane, cell membrane change. And in fact, on the nanometer scale, on the very small scale, what happens is that if you can look at this profile, take some red blood cells from the person, look at it under this quantitative phase microscope, you can get these what are called displacement maps, meaning how much of a deformation there can be in different positions. So these are on the very small nanometer scale, very high precision, like small scale. If you can measure these properties, and that's actually what this is a paper from PNAS from a few years ago. So, so basically you can get these kind of profiles for different types of blood cells, and you can see the profile, and with this tells you something about the disease state of in the case of malaria, for example. Okay, and so like say like you start with this, you have a different some deformation happens to the cell and it comes back. So you look at the kind of displacement profile using this is a quantitative phase images of the red blood cell, and at different locations you can figure out these maps, make a numerical calculation of for different blood cells. What are the properties that are changing? How is the lipid membrane changing? Is there a flickering of this kind of deformation? It's like these are almost like drum modes. Like there are modes of vibration of these red blood cells, and kind of if you beat a drum, there are these modes of vibration of what happens to the drum, right? Similarly, for this donut shape kind of a red blood cell, there are modes of vibration. And is it normal, abnormal? Those are the kind of things you can figure out. So, quantitative phase. So, you have smear of blood cells, red blood cells on a glass plate, and do get this map, get this displacement map. Here, as a function of time, you can get a profile of how these vibrations and shapes are changing. And from this kind of plot, you can compute some statistics and then figure out the disease, kind of say something about the malaria or the disease type in with this approach. Okay, so this is essentially one of the many uses of quantitative phase imaging. So this is one of the applications of quantitative phase imaging. Another application is called multimodal imaging, where you take quantitative phase images. This is an image of a neuron, a nerve cell, and you can study how, for example, if you have two neurons or nerve cells which are coming to each other, they form a connection. So these are called dendrites. Basically, the neurons kind of have how they connect with each other is basically these grow and reach the other neuron, and they connect with another neuron at a point called a synapses. Okay, and there are things like electrical signals that go from one to the other. The strength of the synapse also determines how strong of a signal goes from one neuron to the other, how this forms, how this happens, when in some diseases will this break down, and there is memory loss, for example. So these are the kind of things which quantitative phase imaging, along with this is fluorescence images, multimodal combined images. You do this and you study you can do important studies where you figure out all these properties. Okay. Yes, question. Yeah. Sorry. DAPI is a neuronal, nuclear stain. You are talking about the nuclear stain, right? So T is basically the blue color here is probably DAPI, and the green color is probably GFP, and it could attach to some protein of interest. And so this is a multimodal imaging with different color fluorophores which are added, along with quantitative phase imaging, a combination imaging, multimodal, what's called multimodal imaging. And that so each individual thing tells you some information. Then you can kind of combine information from multiple imaging methods to tell you what exactly is happening. So depending on what you want to study, maybe you want to study how a particular protein is getting transported or something of that sort. So you attach a fluorophore to that protein and look, you quantify some parameters using quantitative phase imaging, then look at how it travels, and so you can, that's the kind of thing that biologists plan experiments, and they use this kind of instrument, work with engineers like us to build, we will give the instrument, they will figure out what to study in terms of biology, and together we can figure out some what is happening in a particular disease and how to make it better, roughly speaking. Okay. Yeah, sure. Sure. So you can look up papers by Professor Popescu. He has looked at prostate cancer, a few other different kinds of cancer. You can with the quantitative phase. It so it's not a perfect technique in the sense that this particular the disadvantage of quantitative phase imaging is that it does not give you kind of what molecules are present or something of that sort, which spectroscopy is another way, which is I'll talk about in a different lecture. That can tell you that. So the advantage of quantitative phase is that it's extremely precise in terms of finding the phase and finding nanometer spatial changes. But chemical changes it does not. So that's why you may use fluorophores, or there may be, so this will give you some information. For some diseases, that may be crucial information. For some kinds of cancer, it may be useful, but it may not be the crucial thing. So that's why you why do if you can if every problem can be solved with one instrument, then why build a second type of instrument, right? This does something well, which is spatial kind of local, kind of height kind of measurement, some kinds of measurements. It does not do some other things, and therefore you need a different instrument to address some other problems. So to answer your question, for different cancers, for some cancers where there are changes in the refractive index in a specific way, he has figured out some quantitative measures where there are some things are changing. He has shown that some things are changing, and that can be measured quite well using quantitative phase imaging. But there are other changes which may not be easy to find, easy to kind of quantify with this approach. So that's where kind of each you build another kind of system which gives you another type of information to combine data and try to figure things out. Okay? I hope that answers your question. Okay. Any other questions? Okay, I think this is a good time to stop for today. Yeah, so in the next class, we will talk about a little bit more on light matter interaction. We'll start with a slightly different topic and different kinds of instruments. So just summarize what we have discussed today. So we started with Fourier optics, and we looked at all the different, a few, the importance of a 4F imaging system, where the concept that with a 4F imaging system, you have access to the Fourier domain in an optical system. By manipulating the Fourier domain in different ways, you can get phase contrast, or you can you can do holography by making using a reference, combining with a reference, or with different approaches. By measuring the magnitude and the phase, you can get quantitative phase images, and this gives you these are all different kinds of imaging systems, which gives you new kind of information and gives you information that you could previously not see. And whenever you invent something of that sort, it is likely that it will be useful for different biological, biomedical, clinical applications, and you can understand something about different diseases. which was previously not possible, and that will kind of and it's a cycle. Then people, like doctors and biologists, will tell you, okay, this is good, but I also want to see this, or I also want, in this disease this is happening. Can you see this? I want to see something else about a particular protein. Something they'll have new, like they'll try this, they'll make some discoveries, and then they'll say, okay, I can see this, and this now, but maybe today I can't see this new thing. Can you help me see it? And then we will try to, we'll use our kind of knowledge of physics and instruments and kind of everything that we are learning today. We'll use our knowledge to build a new instrument which can see this new thing that they are finding it hard to see today. And that's how the world kind of, science and like research works. And you keep going back and forth. Over time, you'll make something which is really useful for everyone. All right, thanks. Yeah.